Thank you. And I will try to give to you an overview of the different type of materials, that is biomaterials and materials that are used for more biotech-oriented applications. And I'm from the University of Mauritius, and just give me a second. Okay, therefore, if we look at what we have, because I like to say that what we are living actually is the materials age. And the grand challenges facing the materials age, they are basically organized, like you have seen this morning, around energy, the healthcare sector, and the agri-tech sector, and the smart factory. And this is where materials science can use powerful tools such as nanotechnology to make radical changes. And since we are from the African continent, these materials age challenges, they are very important to us, and they are all aligned along the SDGs. And SDGs, it's very important to point out, it doesn't only concern the African continent. The SDGs, it's supposed to be a global partnership to address these challenges. Therefore, the Center for Biomedical and Biomaterials Research, we created the center. In fact, academics created the center in 2011, and we organize our activities around building the intellectual capital with training of PhDs and postdocs. We look at particular aspects of research and innovations involving materials engineering, materials blending, in fact. And then we work a lot with industry, where we give to them high-end analytical services. We look at problem solving, and we give also assistance in the uptake of material science and engineering. And today I'm going to try to give to you a brief overview, because I have only 15 minutes, brief overview of the possibilities that we can have using materials engineering. And our main activity when we started, it was organized around making tissue engineering scaffolds, nanofibers and hydrogels for tissue regeneration. That is, we were looking at regenerative medicine. But since we are basically chemists by training, particularly polymer chemists, and chemistry can connect the physical sciences with life and applied sciences. Therefore, capitalizing on that, we have expanded the application, in fact, of polymer chemistry to other areas. And all these areas are extremely important to the region, to the African continent as a whole, but also to our region. The first thing that we looked at was in the health sector. And our hypothesis was that biomaterials can mimic biomaterials in the human tissue. That is, the biomaterials available from land and marine resources could possibly be used to mimic those that we have in our human tissue. And using this as hypothesis and applying science, engineering, and medicine principles to this, we have made wound healing scaffolds, 3D tumor models. We're looking at cancer regenerative medicine. And this is a collaboration, in fact, with the VIDS Advanced Drug Delivery Platform. We are looking at implants for cell encapsulation. We have looked at nanofiltration membranes for antiviral filtration. And we also have a collaboration with industry where we're looking at innovation in textiles. Therefore, starting with materials engineering, there are lots of things that we can address. I'm going to give to you examples in those different areas. The first example will be centered around the healthcare. That is, when I say we are making scaffolds for regenerative medicine, we are trying to look at full thickness skin wounds. And it's not only about healing the wounds, we really want to look about reduced scarring when we are having wound healing. And we've made those nice scaffolds, which are made from piezoelectric nanofibers. Piezoelectric because when we move, it's going to produce small electric currents that is going to promote the cells to grow, proliferate, and form new tissue. And we've been able to come up with scaffolds that can resolve scar formation. Therefore, if you look at those two images, these are from our in vivo studies, you'll see that here we have scar formation on here on the control without scaffolds, or if we use a positive control, that is commercially available scaffold, and with the piezoelectric nanofiber scaffolds, we have no scar formation. Therefore, this is a very interesting study that is continuing because, of course, we want to have a human clinical trials and continue until we can come up with a real application. The second thing that we've used for skin regeneration, they are based on hydrogel. The first example was using piezoelectric nanofibers. Here we have a self-healing injectable scaffold. Injectable scaffolds, they have the advantage of addressing non-uniform wounds because you can't preempt on what will be the shape of a wound. Therefore, if you have an injectable scaffold, it's much easier to work with because it's a hydrogel. It's going to take the shape of the wound. You inject it, it solidifies and takes the shape of the wound. And we've got 3 generations of scaffold. When we start off, A here is the first generation, B is the second generation, and C is the third generation. And C is really a clean wound healing that we saw. And it's not only the picture of the wound, but we have to see what happens underneath the wound, because why we're doing this skin regeneration as well is because we want to tackle the problem of non-healing wounds such as diabetic wounds. And most of these wounds, they are non-healing because they open up after some time. And why do they open up? It's because of the weak mechanical properties of the newly generated tissue. That is why we need to look at what is happening underneath this nicely no scar formation wound healing. And if we look at C here, what's the difference between C and the others is that C, we really have a mature tissue formation with strong mechanical property, which means that it's not only no scar formation, like here, this, but it's also the quality of the tissue regenerated. D and C, one is a positive control here, that is we have used collagen gel because collagen are very famous for giving rapid wound healing and it's a commercial product. E is a negative control. Even the commercial product will generate a scar. And we are now applying this particular scaffold, hopefully in our first clinical protocol. We are first looking at cosmetic applications, and we are looking at how to use them as transdermal subcutaneous injections. And that was skin. But if you have scaffolds available, you can look at different types of tissues. Therefore, the next tissue that we looked at was the bone tissue. And bone regeneration, as you have seen earlier, we were talking about the aging population, the need for bone scaffolds. Bone regeneration is a very complex process, and we have tested the scaffolds in bone regeneration. And this is a collaboration with our center and the CIWA Center, which is found, Mauritius is here. CIWA is in Réunion Island. Réunion Island is a French territory, and we did this preclinical trial under an EU-approved protocol in an EU-approved animal facility level 2, and we looked at how we can reconstruct the skull after a wound. And if you look at what we could generate, this is our hydrogel, the injectable hydrogel that I showed to you for skin. We used it for bone as well, and we could see rapid bone formation. In fact, when this is just the first studies that we did, you can see that the bone regeneration is so rapid that it becomes uncontrolled. But now we have other results where we have controlled this process. And the good thing here is there's good fusion between the new bone and the bone that was here previously. And if you don't have this good fusion, and it's going to affect this area in the skull. And here you have the negative control, that is no scaffold. You just allow the skull to regenerate itself. And here it's a commercial product that we have used. This is the commercial product, the positive control. And it's a collateral medical device containing hydroxyapatite. And hydroxyapatite, you can detect it in the CT scan. That's why you can see it at week 2. This is the material that was put there at week 0. But the thing about it is that you don't have a proper fusion between the newly formed bone and the bone that was there previously. And this creates sites that are prone to mechanical weakness, if I can say. Now, we didn't stop there. I had a very interesting conversation with Professor Bolton from UCT. And in fact, he's the one that germinated the idea in my head about why can't we use coralline algae biohydroxyapatite for the piezoelectric scaffold. And we did that. The study is under review. We, in fact, extracted biohydroxyapatite. Hydroxyapatite is an important constituent of the bone, and we have grown bone cells on them, and they're giving to us great results, because for biohydroxyapatite to give good results, the CAP ratio that is formed should match that of the human bone. And here you can see very nicely growing bone cells on the scaffolds that we have generated using biohydroxyapatite from coralline algae. Therefore, we can make use of a number of biomaterials from land and marine resources, because Mauritius is just a small island, but our exclusive economic zone, our marine area, is huge. And exploiting materials from the marine environment is very important to us. And in the marine natural products, we've also looked at a number of molecules, because we have identified a molecule. I'm not talking about extract here. I'm talking about a molecule. And this study has just been published. And again, this is a collaboration with the WITS Advanced Drug Delivery Platform. And the study has been published. We have 2 more manuscripts under preparation where we are using this molecule in scaffolds and addressing cancer, because it has got anti-cancer properties, and some other molecules have got anti-diabetic properties. Therefore, again here, making use of materials engineering to be able to address healthcare issues. And rapidly, because I think I have 4 minutes left, we have readapted our scaffold to look at what we coin as cancer regenerative medicine. You know, when you remove a tumor, there is a wound which is formed. Now, since we have tested these wounds already for skin regeneration, why not use it to heal cancer resection wounds? But the tricky thing here is that you don't want to provide a scaffold to the remaining cancer cells so that they can regrow and make the tumor come back. Therefore, this scaffold, they are very specific. You have to choose your materials very carefully so that you have the good cells growing and the cancer cells not growing. And this manuscript has been submitted and it is under review. Again, we collaborated for the in vivo studies with WITS. And 2 last things on in the medical area is we can generate tumor models. That is, we've got spheroids that we have generated so that we can mimic the tumor in its natural environment. And what is this used for? It can be used for drug screening. Therefore, instead of having a flat culture of cancer cells, which does not mimic what is inside the human body, using hydrogels, we can grow spheroids like this, you can see, with different cells, because in the body, it's not only one cell, it's different cells, and then we can do drug screening on these. And lastly, we are looking at encapsulating cells in our hydrogel from the pancreas for subcutaneous implants for insulin production. And this is an ongoing collaboration with Max Planck Institute for Medical Research. I mean, this would be fantastic if you can have an implant under your skin with cells in it that can produce your insulin. Now, with all this data, we got into machine learning, and we are modeling, in fact, the activity of our materials, of our scaffolds, and we want to predict the biological process so that we can go faster. We don't have to spend time to do 200 materials blending, 200 in vitro testing, then 200 in vivo testing. We don't want to spend time like this. Now we are using our data to build models so that we can go more rapidly into making the appropriate choice for our materials. Now, using all this knowledge, we have the knowledge on materials. During the COVID, we came up with a nanofiber filtration membrane. It has been certified and tested with a 99.95% of antiviral efficacy under increased challenge, and you can wash it 20 times and reuse it. We have tested it. And this is our collaboration with the textile industry in Mauritius. And we've gone further now. We are testing high-performance compression wear. And with athletes, this is an ongoing study so that you can improve the performance of athletes. Therefore, you can see that using materials, we can have a wide variety of application. And the last application that I want to show to you is in the agri-tech sector. We've got a team of 4 groups working on it, and we are looking at nanofertilizers and nanovaccine, and our part does not change. We are here, we are in materials engineering, and then we have collaborators with different expertise that we are bringing together. To finish off, we organized last year a talk for how we can have a sustainable biotech for the African region. And we got the honor of having Prof. Langer. He is behind the creation of Moderna vaccine. And what came out of this discussion is that to have a regional biotech industry, we really need to build on our human capacity. And we really need to network labs. This is very important for sustainability. And what will be the timeline of this? What will be the cost involved in this endeavor? These are important factors that we need to consider. Therefore, possibilities for our Arua Centers of Excellence is really to support material scientists and engineers and support frontier science, engineering, and medicine, create networks, create co-chairs, and all this work would not have been possible without a team. And I'm very proud to say that the team is 70% women scientists. And so we can't say that we have a problem of attracting women in materials engineering on our side. With this, thank you. And I think I've kept my time. Thank you.